Dear student, today we will discuss anatomy of flowering plant. In the anatomy of flowering plant, we need to understand what is plant tissue system. The plant tissue system is organized in three different categories. You can say dermal, ground tissues and vascular tissues. Dermal tissues usually present on outer surface and provide protection, and they basically consist of two different types of tissue, epidermis and periderm. Epidermis occur during primary growth, whereas periderm during the secondary growth. Then ground tissue, they consist of all the tissues of the plant except for the epidermis and vascular, and composed of parenchyma, collenchyma and sclerenchyma cells. Then vascular tissues are the conducting tissues and they conduct food and water. We'll be discussing them again, but before that we will be discussing what all different types of cells are there in the plant. So the plant growth is not limited as in animal if you compare it with the animal. Therefore, new cells keep on adding and old cells start differentiating into different cell type and form plant tissue system. So the new cells that get added, they are called meristematic cells, and the old cells which keep forming from the new cells are called permanent cells. So if you see, the plant tissue is divided into two categories on the basis of origin and differentiation: meristematic and permanent. Meristematic, they are undifferentiated actively dividing cells usually present in actively growing region of plant. Permanent, they are differentiated. So we'll see what are they. First we'll discuss the meristematic tissue. So three different types of meristematic tissues are there at different location: apical, intercalary and lateral. The apical is also known as primary meristematic tissue. Intercalary is also known as primary meristematic tissue, whereas lateral is called secondary meristematic tissues. Apical usually present at shoot and root tip and at axillary bud. So if you look at the position, this is alice of shoot apex. So here where they are located, shoot meristem. And if you look at the root region, just between the root cap and the root where there is the origin of, you can say, root is there, apical meristem lies in this particular portion. So axillary bud usually at the axil of the leaf. Okay, and if you see the meristematic, intercalary meristems, they occur at the base of internode and also at the base of leaf sheath in monocots. In grasses, these tissue help to regenerate plant that is removed by grazing of the herbivores. Lateral meristem, they are called secondary meristem. They are present in the mature region of root and shoot and are responsible for producing secondary tissue for secondary growth. So in plant you have primary growth and secondary growth, which takes place in the later on stage, which is responsible for increasing girth of the plant, is called secondary growth. Now lateral meristem, they are parallel to the long axis of stem and root. For example, fascicular cambium. So if you look at here, you can see the position, the apical meristem at the shoot tip and root tip. Then the lateral meristem, the increases girth and they are present in the parallel form and throughout the plant. And then intercalary, which is at the base of internode. Okay, so the root and shoot meristem grows and form three undifferentiated cell type, which are protoderm, procambium and ground meristem. Protoderm later on give rise to epidermis. Procambium give rise to vascular bundles and pericycle. Ground tissue give rise to cortex and pith. Now we will see that what are these cells and how are they arranged in the plant. The plant growth occur where meristematic cells are present, which form the region of cell division. The newly formed cell grows. This forms the region of cell elongation. And finally, the cell differentiate into different cell type to carry out a specific function. This region is called the region of cell differentiation. Now, moving on to the okay, so here just we'll just go through the how they are located. So you can see the root tip and the root region. So if you see three different zones are there: zone of cell differentiation, elongation, and cell division. Okay, so cell division where actively these meristematic cells are present and the division is occurring, and just above the root cap is the slow dividing region which is called quiescent center, and this is surrounded by a layer of rapidly dividing cell called cleptogen. Now you can see that these cells give rise to the cells of elongation and further the cell of differentiation. So if you look at this, it is divided into three different regions: the procambium, which is in the center region, which give rise to the cambium, and sorry, also the vascular tissues, the ground meristem, which give rise to the cortex tissue, pericycle tissue, and protoderm give rise to epidermis. So similarly, you can see the formation in the uh, ls of shoot tip. Zone of cell division, it's on the top, then elongation and differentiation. So here also you can see protoderm, procambium, okay, and ground meristem. Okay, so here you can see these meristematic cells. Now if you look at the permanent tissue, then permanent tissue, they mature, they do not divide, are divided into two categories: simple and complex. The simple one made up of one type of cell, whereas the complex made up of more than one cell. Okay, so simple are parenchyma, collenchyma, and sclerenchyma. So we'll just compare the three. They are living cell within the cell wall. Collenchyma are also living cell, but their cell wall is thick at the corner, and sclerenchyma are dead cell with thick cell wall due to deposition of lignin. Now, in the collenchyma, the deposition at the corner is of cellulose, hemicellulose, and pectin. Here there is no deposition. They are isodiametric, means same diameter, and closely packed with intracellular space. So you can see nucleus clearly. You can see the vacuolar very prominent. You can see cytoplasm, and this is how they are arranged with little intercellular space. Now collenchyma, they are elongated and closely packed with a no intracellular. So there is no, because of the thickening, you cannot see any intracellular space. Okay, and this thickening region at the corner is because of the deposition of pectin. Now collenchyma are absent in monocots as they have early development of sclerenchyma, which are already hard and they provide the mechanical support. So if you look at the sclerenchyma, they have deposition of lignin. Lignin impart the tensile strength and waterproofing quality, and they are polygonal without intercellular space. They are of two types, sclerids and fibers. So if you look at the sclerids, they are isodiametric as compared to the fibers, which are elongated and pointed at the end. Okay, and sclerids, you can see the thick deposition of lignin over here. So this is how these three cells are there, which are simple cells. Now we will see what are the complex one. The complex tissue are basically xylem and phloem. So the xylem form the continuous channel through the root, stem, leaves, fruit to conduct the water, and it consists of four different things: tracheids, vessels, xylem, parenchyma, which are also known as wood parenchyma, xylem fiber, or they are also known as food fiber. So tracheids, they are dead, vessels dead, xylem parenchyma are the only living one. So tracheids, they are single and elongated cell, whereas if you compare them with the vessels are formed by the fusion of several cell by dissolution of the end wall. So they form a tube-like structure. Then tracheids have tapering end, whereas the vessels have round ends, and tracheids are non-perforated, so water move through bordered pit. Whereas vessels are perforated. Tracheids, they are predominant in gymnosperm and has narrow lumen, whereas the vessels, they are dominant in angiosperm and they have wider lumen. Now, the xylem parenchyma, they form the radial sheet of tissue called medullary rays, which is meant for the conduction of food and water. Xylem fiber consists of sclerenchyma and they do not conduct water but provide mechanical support. Now, if you see the phloem translocate the prepared food material from leaves to other part of plant, and they consist of four different types of cells: sieve tubes, companion cells, phloem parenchyma, and phloem fibers. So, out of four, three are living, whereas one is dead. Okay, that is the phloem fiber because they are made up of sclerenchyma. Now, sieve tubes, they are long tube formed by end-to-end fusion of the cell, and the transverse wall is perforated like sieve. Companion cell are elongated cell and connected to sieve tube by simple pits, and they help in maintaining the pressure gradient in sieve tubes. Whereas phloem parenchyma, they are tapering cylinder cells and they store food material and are present in the dicot only, and they store resin, latex, and mucilage. Phloem fiber, they are absent in primary phloem and occur in secondary phloem, and they have thick cell wall at maturity and lose the protoplasm and die as they provide mechanical support. Okay, so now we'll see types of xylem based on when they are formed. They are divided into two categories: primary xylem and secondary xylem. The primary xylem formed during the primary growth of plant from procambium. The xylem parenchyma are less extensive in primary xylem, whereas in secondary xylem, this is formed during the secondary thickening from the cambium, and this is here the xylem parenchyma are more extensive. Now again, primary xylem is of two type: protophlloem and metaphlloem, depending on their position where they are forming. So protophlloem is the first form of xylem occur at the in the apex region, and they are crushed by the surrounding tissue during the secondary growth. Metaxylem, later form of the xylem, occur in mature region and they are functional for long time till secondary thickening. They are present throughout, they are functional throughout the um, life of monochord. So similarly, we have different types of phloem. Now, depending on how the xylem is arranged, the condition is called endark and exarch. So, how the different meta and protoxylem are arranged. So, if you see the endark condition, the protoxylem is located toward the pith or the center, or you can say toward the inner side, whereas metaxylem is located toward the periphery or exterior. So usually such kind of condition is seen in stem. If you look at the, this is called endark. If you look at the other condition, which is called exarch, in which the protoxylem is located toward outside, whereas the metaxylem is located toward inside, and usually this is seen in the leaf. Now we'll just discuss the tissue organization. So here you can see the dermal tissue, which consists of epidermis and periderm, the dermal tissue. So epidermis you will see in the during the primary growth, consist of basically parenchyma cells. They are arranged without intercellular space in the epidermis, and here Some of the epidermis are also modified to stomata. Mainly function is the protection from heat, cold, and pathogen. Also exchange of gases. Cuticle, which is present just above the uh, you can say epidermis, it checks the evaporation. Okay, and then we have, if you compare the epidermal hair in shoot and root, you will find that in root they are unicellular, whereas in shoot they are multicellular. In unicellular, they help in the absorption of water and mineral, whereas in shoot the epidermal hairs consist of secretory function. So there are two different types of modification that are seen. One is lacticiferous and glandular. So lacticiferous, they produces lactase, which is a secretory product, and glandular produces honey, oil, resin, poison, etc. Okay. So in root, the epidermal hairs are called epiblema, whereas in shoot they are known as trichomes. Okay. So the epidermis itself is called epiblema, and here it is called the trichome. Okay. Now if you look at the periderm, which is again a dermal tissue, so again the function is protection, and it is formed during the secondary thickening function. Okay, and it consists of three different types of tissues: cork cells, secondary cortex, and cork cambium. The cork cells, they are dead subrised cell that bears lenticels, which are on the outer surface. Secondary cortex is toward the inner side, and cork cambium, which is the middle layer, formed from the subepidermal layer in the stem and from the pericycle in the root. So this is regarding the uh, dermal tissue, and in the next video we will be discussing certain other tissue and the secondary thickening. Thank you.